I'm sorry, what was your question?
I didn't catch the name of this bacteria.
I don't think he said the name of the bacterium.
And I had a question.
Sure.
So they were talking about if needed, they'll send a large concentration of these bacteria into the water.
Right.
What effects would it have on, like, fish, the marine life, like the birds? What would happen if these microorganisms begin to reproduce and we'll have an overpopulation of this bacteria,
Well,
such as we do for algae?
The overpopulation of this bacteria is going to be a good thing because it's going to eat the oil.
Okay. The food's the oil, so if
The
the
food is
source
the
isn't
oil.
there,
I was gonna say,
Once
once
the
the
oil is
oil's
gone, then they run out of food. They don't have another source. So it's really a very good mechanism. But
if
like,
they're eating lipids,
huh?
if they're breaking down the lipids that's in the oil, wouldn't they be able to find that in fats and, let's say, like the...
Not the same kind of, it's not the same kind of hydrocarbon. So these are specific to the hydrocarbons that are found in petroleum products. Yeah. So they're not ones that are going to cause infection in animals or anything like that. They run out of oil, they die. And the nice thing with the good thing is they're very useful for cleaning up shorelines because they have a substrate to kind of attach to. But like they said, out in the open ocean, they're not so sure how effective they really are because the ocean is so big, and they would probably diffuse outward, and you might not have them concentrated where all the oil is. So they're really a very good way of cleaning up the oil spill on the shoreline, but they're not so sure how well they would do out in the open ocean. So.
Or like other organisms, wouldn't they go to the nutrient supply?
No, they're too small. I mean, a microscopic organism, they're not going to, in a big, huge open ocean, I mean, they do have chemotaxis, but probably not to that extent over miles and miles, because, you can be swept away very quickly in the ocean, so they don't have that kind of capability. Their little chemotaxis occurs on a microscopic level. So, all right, a couple other things about breaking down the oil. We've been doing this for a long time. Since the 1970s, we've been using microbes to help bioremediate oil spills. One of the major times it was used was in 1989 with the Exxon Valdez, which happened off the coast of Alaska. And unlike the oil spill in the Gulf, the BP oil spill, this really was concentrated along the shoreline, and so they were able to really use the microbes to help clean it up. I do have to go back for a moment to the biofuels, because I forgot to tell you a couple of interesting things. So when we were talking about biofuels, we were talking about how they use corn. And there's been some problems with that because it takes a lot of area to grow corn. It also is a human food, and so by using it to make fuel, we're denying, food to people, plus we're jacking up the price of corn because it's so in demand. So there are thoughts about switching to using something called switchgrass. Switchgrass is a grass. It requires less space to grow it, and it's not a food source. So it's not like, we're going to drive up the cost of a food source and have a food source be in limited supplies by using it. So switchgrass. And places like Brazil, they don't use corn or switchgrass. They use sugarcane to produce their biofuels. So that I forgot to tell you, that's that. So switch pages
They
again.
don't have the same problem. They don't have the same problem with
You
sugar
would think
cane
so.
that
You
we
would
do
think
with
they
corn.
would, because I would think
It
that
still
would
takes
take
a lot of space to grow,
and
and
it's
it's a
also,
food.
yeah.
It's the top part of the plant that isn't gonna be turned into sugar.
Oh,
And there we have it.
okay. That's
If they-
perfect.
Like, same thing,
So
you
it's
could
using
use
like more of
corn
the,
stalks that you're
Yeah.
gonna turn into, which right now they plow back into the field. If you
Yeah.
can figure out a way that there's not a lot of energy in a dried corn stalk.
But what there is in the sugar cane. Interesting. I learned something new every day too. Yeah.
I took nutrition, I was told that we don't really use sugar cane as a source of like the sugar in our foods that much. We actually use beet sugar as well.
Beet sugar? Really? Well, that's interesting too. See, I learned two new things today.
Well, 3 things because of the slide thing too.
That's right, 3 things. Gosh, 3 things. Okay, I'm done for the day. I've learned my 3 and I'm good. All right, so moving on with bioremediation, the next video, and I had the same problem with this one, by the way, so I might have to just do copy and paste for it. But we can also use things like fungi to bioremediate. And there's a microbiologist, specifically a mycologist. Mycologists are ones that study fungi, by the name of Paul Stamets, and he's a very famous mycologist. He has a TED Talk. If you ever get bored and you want to watch a TED Talk, he has one called 6 Ways Fungi Can Save the World. If you are any of you bio majors? No? Come on, be a bio major. If you become a bio major, you'll actually watch the whole TED Talk. It's pretty interesting. We have a very short excerpt from it, and I do have to say it's not a very good excerpt. I don't know who filmed it. It wasn't the TED people. It's kind of dark, I think, but listen to it. Right? It's really interesting. And he talks fast, by the way. It's very interesting what he can do with fungi to bioremediate. So let me see. I'm going to probably have to do the same thing I did with the other one. Nope, that's
This
not
is the Gulf of Mexico.
it. Hold on. It's the first one. Even though the first one is what I clicked on to watch that other video. I don't get it. I don't understand why it does this. I must have changed the video.
Modern civilization is addicted to oil.
This isn't the video that I thought. It must be on the next slide. This must be a new one. I added a couple of new videos this semester, like that. The corn one that you guys liked was actually a new one. So this might be a new one. I don't remember what it is.
Or otherwise consume some 90 million barrels of the goop every day. That ends up making a mess. According to the U.S. Environmental Protection Agency, there are an average of 70 spills every day, mostly insignificant. Every once in a while, we make a spectacular mess, like the punctured Exxon Valdez oil tanker off the coast of Alaska, or BP's blown-out Macondo well at the bottom of the Gulf of Mexico. When humans make a big mess like that, humans try to clean it up. We corral it with booms or burn it off, but whatever we miss ends up getting eaten by microscopic bugs. In fact, bacteria and other microbes are one of the main reasons that BP's Gulf oil spill wasn't a bigger disaster. A big bloom of bugs ate most of the 4.1 million barrels of oil spilled before it could wash into
I'm still
wetlands
skeptical.
or tar sea creatures. How big was the bloom? 100 sextillion microbial cells big. Here's how they work. Oil has been a part of life on Earth for eons, and microbes evolved to take advantage of that. Tiny bacterium feast on the oil and gas seeping from the bottom of the seafloor or bubbling to the surface. They're not necessarily fast, but they are thorough, turning loose hydrocarbons into more microbial cells. But microbes don't only eat oil. Plastics, often made from oil, get everywhere too. For example, the Great Pacific Garbage Patch is
Oh, I
made up
did
of
get a
tiny
video
grains of
about
plastic
plastic.
and the occasional grocery bag. It's actually proved to be a hospitable home for plastic-eating microbes and other microscopic life. This new ecosystem has been dubbed the plastosphere. On their own, microbes would probably take eons to clean up our plastic mess. But with synthetic biology, we could tweak these little friends and help speed up that process. We could help them to help us. Scientists have been working on souping up microbes' ability to eat oil for decades. The very first genetically modified organism to be patented was an oil-eating microbe way back in 1980. There's no reason that plastic eaters couldn't be enhanced in the same way. It's obviously better to avoid trashing plastic and dumping oil in the first place. But since we're a messy bunch, It's a good thing we have microbes around to help us clean up. For Scientific American's Instant Egghead, I'm David Biello.
Okay, so I did put one about plastics on. I upgraded or updated my PowerPoint in, I guess, early January. I guess I forgot I had done that, so that's good. All right, let's go on to the next slide. All right, this is the one with the guy that talks fast, where it should be. There we go.
So the other project that I've been doing for well over 10 years is neutralizing petroleum-based hydrocarbons using mushroom mycelium. And this is a project that we've done that's been written up now in several journals. This is diesel, and this is mushroom mycelium. And this diesel plug, the mushroom mycelium, is absorbing the diesel, it denatures it, and the mushroom mycelium produces these extracellular sweat beads, again, enzymes, lignin peroxidases. And the lignin peroxidases break carbon-hydrogen bonds, which is the bond that's the primary bond in lignin. Well, that's also the primary bond in hydrocarbons. So they're exquisitely well designed for breaking down a whole suite of hydrocarbon-based contaminants. And so we have now challenged a whole bunch of strains against a whole bunch of contaminants, and we found excellent results in breaking down PCBs, PCPs, dioxins, virtually all pesticides, most herbicides, all sorts of petroleum-based fuels, etc., diesel, gasoline, etc., etc. So the Department of Transportation in Bellingham, Washington, was being fined by the Department of Ecology, one state agency fining another state agency, treating it just like we would treat another individual or corporation. That's the way the law reads. Because they had a diesel contamination spill in a big transportation yard where all these buses were being stored. And because the toxic waste, the levels of these PAHs, which is polyaromatic hydrocarbons, was so high, this was off-limits facility. You couldn't do any activity there. So they invited 6 bioremediation companies, and so we had a little contest up in Bellingham, Washington, for who could break down this diesel-contaminated soil the best. And so there were 6 piles that were created, and we had the enzyme chemical treatment people come in, we had the bacterial people come in, and we had other type of systems that were employed to break down these PAHs, these diesel compounds that are present in the soil. And so we inoculated our batch, and then we looked at the piles 4 weeks later. 4 weeks later, a group of scientists and government officials went up to Bellingham, Washington. They pulled apart the black tarps. The first tarp was pulled back. It was black and neutral, diesel-smelling soil, stinky, dirty black soil that was just stifling in its odor. Second pile, same story. Third pile, same story. Fourth pile, same story. The 6th pile, they pulled out, and ours were covered with oyster mushrooms. Okay, all right. And some of these oyster mushrooms were up to over 12 inches in diameter. Now, any of you collecting oyster mushrooms realize that this is a testimonial to a happy mushroom. The fact that these mushrooms are so large. So we hit a home run. Now several other things happen. I know you have to stop here in 5 minutes. I have 2 more slides. The mushrooms then, after about 6 weeks, 7 weeks, began to rot. The mushrooms produced spores. Flies were attracted. The flies laid eggs. Larvae began to be produced. Birds came in after the larvae. The birds brought in seeds. The seeds germinated. Plants and weeds started to grow. I don't have the final photograph, but after 8 to 10 weeks, our berm was an oasis of life, a green zone. And so we invited bacterial remediation because the mushrooms rotted. We invited phytoremediation because the birds brought in seeds. So I believe we found a keystone mechanism by which we can cause a domino effect of remediation that can break down toxic waste.
That was it. He ends quickly. Okay, so he talks about how they have this pile of biodiesel contaminated soil, He inoculates it with mushrooms. He calls what were they, oyster mushrooms, did he call them? Oyster mushrooms. And what did he say the oyster mushrooms did to the oil? They broke it down, right. But that's not all they did. What happened next? They died off, right?
Yeah, they decomposed.
The flies came because there were mold spores. The flies came, well, actually before the flies came, ate the mold spores. Flies, when they ate the mold spores, they laid eggs. So now you have flies and you have maggots. What came next?
Birds.
Birds.
The seeds.
And birds, how would they bring seeds? Yep, they would go, they would drop the seeds after they went through their digestive system, and then the seeds would grow, and now we have this nice, happy little pile of dirt that's growing stuff. Once you have stuff growing, now you not only have the flies, you have the birds, and then you're going to bring in animals eventually and so forth. So it turned out to be a really good way of remediating. All right, so this one is your second video that's homework to watch. So I'm going to show you how to get to it, just like the other one that I know some of you have already watched. Make sure that you watch the other one as well. But you click on learner.org. Come on, there we go. And then you go down here to science, and then across to college/adult. Click on it. Then you scroll down near the bottom. This is the only one that's going to have the word microbiology in it. So it's Unseen Life on Earth, an Introduction to Microbiology. You click on that. You scroll down, and the one you want is number 8, which is microbial ecology. And you just click on this video on demand button right here, and it'll bring it up. They're about 20 to 25 minutes long. So you've got the one, this one, plus the other one, which was the what was the other one? Microbial metabolism. Okay, so you have them to watch.
Number 3 and number 8.
Yes. All right. I'm not going to ask this question yet because you haven't watched it. Let me pull up the next video. Or it's not video. The next PowerPoint.
No, you're fine. I was following up that last time.
Okay, so we're going to get started just for the next maybe 5 or 10 minutes talking about microbial growth. And when we talk about microbial growth, we are not talking about the growth of one individual cell. Microbial growth really refers to increase in number of cells. So it's really more of a growth in population, not in size of each individual organism. Okay? So in order to talk about population growth or microbial growth, we have to first know how bacteria divide. And they divide by a process called binary fission. Basically, with binary fission, the bacterial cell is going to elongate somewhat. It's going to replicate its DNA, so there's 2 copies of DNA in the cell. Why does the bacterium copy its DNA so that there's two copies? Right, so when the cell divides, each new cell has a copy of the DNA. Once the DNA's been replicated, the DNA starts to kind of separate itself somewhat to different sides of the cell. And then what you see is the cell wall and the plasma membrane start to invaginate and start to pinch inward. And eventually that's going to form a cross wall, which is going to completely separate the 2 cells. And eventually they're going to be completely separate. So that's binary fission. I do want to mention a process that's kind of similar. This is, by the way, this is a form of asexual reproduction. Right? Form of asexual reproduction. Another form of asexual reproduction that's important with microorganisms is one called budding. You don't see it very often with bacteria. It's more something that you see with yeast. Again, yeast are microorganisms, but they're not prokaryotic. They are eukaryotic fungi. Okay? But when yeast carry out the budding process, which also is asexual, again, they're going to make a second copy of their DNA. And then what happens is, here's your yeast cell. You have this little bud form. Okay? that's gonna contain one copy of the DNA. This parent cell is gonna have one copy of the DNA. And then what happens is that bud is gonna get larger. Right? Until eventually it's the same size and it separates off, okay? So it's different, whereas binary fission is a very equal distribution of the cytoplasm. With budding, it's not an equal distribution. There's a very small amount of cytoplasm that buds off, and then that starts to grow kind of independently, but while it's still attached. Okay? So that's just another form of asexual reproduction. So I want to talk about generation time. Generation time can also be called doubling time. And it's just the length of time it takes either for a cell to divide or for the population to double. Okay? So the average generation time for bacteria is between 1 and 3 hours. However, that does vary quite a bit. Something like E. coli has a generation time of 20 minutes. That means every 20 minutes, E. coli divides. Something like Mycobacterium tuberculosis has a generation time of 24 hours, right? So that's much longer. So we can look at this and we can see generation number and then number of cells, and then we'll talk about this in just a minute. So generation number, the first generation is going to be 0, and that's where there's 1 cell. And after 5 generations, you have 32 cells. So how does that happen? So we can make up our own little chart. Generation number, we have 0, 1, 2, 3, 4, 5, and we'll have number of cells. And after 1 generation, how many cells do we have? 2. After the second generation, we have 4. After the third, 8. Because those 4 cells are going to divide.
And then 16,
And then 16,
32.
and then 32. So you can see how it fits in with this 0 and then 5. What you're going to notice is after about 20 generations, you're going to have over 1 million cells. And if this were E. coli, right? This were E. coli with a generation time of 20 minutes, if generation 0 is at time 0, when are you going to have generation 1? 20 minutes later. How about 2?
40 minutes,
40,
an hour,
1 hour,
an hour and 20.
1 hour 20, 1 hour 40. So in an hour and 40 minutes, you go from 1 cell to 32 cells. Now, let's say this were Mycobacterium tuberculosis in the time, and I'll move out of the way as soon as I write. I'm sorry, I'm kind of backing myself into a corner. Remember, their generation time is 24 hours. So to go from 1 cell to 2, it takes 1 day, 2 days, 3 days, 4 days, 5 days. So you can see, to get the 32 cells of Mycobacterium tuberculosis is going to take 5 days versus E. coli is going to take an hour and 40 minutes. Huge difference. That's why in a laboratory setting, if you're going to grow mycobacteria, you're going to hold those cultures for many, many weeks because the bacteria grow very, very slowly. Okay? E. coli grows like that. I mean, it grows super fast. Right? Oops. So let me go back just one more, just for one thing. So you'll notice on the right, log base 10. So a lot of times with population growth, we graph it in log base 10 instead of the actual numbers. And so if you look at the next picture, if we graph the actual numbers, we get what's called a J-curve, right? But if you plot the log base 10, you end up with a straight line that indicates population growth, right? What generally will happen though is there's going to come a time where the organisms might run out of nutrients or space or something like that, and then they'll reach what we call carrying capacity, and this curve will actually flatten out. So if it's a J curve, it becomes an S curve. If it's log-based, it'll just go straight across, and that's when the population is no longer increasing. And we're going to talk more about that in the next class when we get to this, the growth curve of microorganisms. So that's where